Hello, this is a recording uh, of some lecture material. Uh, some of it may be a re-presentation, but you should take a quick look at it in preparation for the dissection on Wednesday. And it involves the central, and basically we're dealing with central nervous system anatomy, and most of the information has to do, and it's really the way it's set up. So effectively, originally in embryology, the development of the brain, the advanced portion of the brain is called the forebrain, and it goes by these fancy names telencephalon and diencephalon. The telencephalon is really the highest functioning. That's the where your gray matter mostly is located in the cerebral cortex, and the cerebral lobes, which is really the highest order of functioning. And the diencephalon plays a pivotal role in organizing and distributing and reacting to the material. And effectively, those are the highest order of function. The midbrain remains the same from when your brain starts to develop during gestation in utero. And the midbrain basically just keeps the name midbrain or mesencephalon, where the hindbrain are lower order functions, as to some extent as the midbrain, which really deals with things we don't even think about, unconscious things, regulating heart rate and blood pressure and breathing and just maintaining the inside, making sure blood flows properly and how your blood vessels are functioning at any given time. Most of the things that are basically taking care of themselves. And that's what I really wanted to go over with you in this little presentation. So the cerebrum or the cerebral hemispheres and everything more or less has a right and a left, with the exception of when we start to get down a little bit lower into the brain stem. And the brain stem area is defined as the midbrain, pons, and cerebellum. There's a very interesting area. Beg pardon. The brainstem is defined as the midbrain, pons, and medulla oblongata. The cerebellum is very interesting. It's a unique area that sits sort of behind the brainstem and communicates with it. And what it does is it coordinates all types of muscle activity. And so anything that your muscles do is a coordinated effort. Even when you point your finger, you have to move certain muscles in a certain way. When you stand up or sit down. So all of the muscles, and particularly those on the same side, have to be organized to do whatever the activity is. The fancy term that we give to that is called ipsilateral. Contralateral means on the opposite side. It's like the right side of your brain controls the left side of your body. Whereas ipsilateral means things that are going on the same side. So the cerebellum, like I said, when you point your finger, write something, all of the muscles that you're using, whether you're right-handed or left-handed, are being organized and that activity remembered, so to speak, by the cerebellum. And interestingly enough, all of these structures are basically surround a hollow tube. And this hollow tube is called the neural tube. There's fluid within it, centrally placed within this tube, as a lot. Most hollow tubes are in the body, have some kind of fluid activity. And as the brain begins to develop and enlarges and bends and shapes itself, this tube has some very, very interesting anatomical appearances that you will see. And the fancy name we give to these hollow fluid filled cavities, and yes, they're filled with something called cerebrospinal fluid, abbreviated CSF, that is so terribly important. So here you can see it starts with this hollow tube, and you can just start to see it around the edges, the brain matter starting to develop. And so consistently, the pattern we see in most of these areas is we begin to see fluid filled tube, a little bit of gray matter, and then effectively what you're going to see is a lot of white matter on the outside. There's two areas that we call cortices. Remember, cortex is for outside, medulla is for inside. So there's two areas where we refer to as the cortex, which have an additional layer of gray matter on the exterior. And this is really the highest order functioning stuff that we have. And so there's some in the cerebral hemispheres we call the cerebral cortex, and we're going to go over those extensively. And then also the cerebellar cortex. Those are really the two areas where we have the highest. So you're going to see this repetitive pattern. And we name the ventricles, as you'll see this very interesting diagrams that come up, by what they're associated with. So the cerebral cortex, the ventricles, are sort of funny looking shape. Looks like a letter C, and they're connected. And we used to call them first and second, but they really couldn't make a determination whether right or left were first or second. Now we call them lateral ventricles. And then another one that's in the middle of what's called the diencephalon, a really important area, again, coordinating information and shuttling it in the right direction, processing and pre and post processing it, and giving rise, as you can start to see here, to your visual activity. These are the starts of the optic nerve. And because we process so much from what we see, I mean, more than half your brain is dedicated to processing visual matter. We're very visually oriented. If someone has poor sight or loses their sight, then other senses like hearing and touch become much more, and smell, all those become much more acute as they begin to develop. That's what the body does, it compensates. And see, here's the midbrain, which again is mesencephalon. Mes means middle, and it's part of the brainstem, as is the metencephalon or pons, or the myelencephalon, which is the medulla oblongata. But exterior to it, that cerebellum is kind of important. So you can see the names of these ventricles, and we'll look at them more closely. So since the brain grows so fast, it folds and folds and folds and folds and folds and occupies space. So everything moves toward the brainstem. It gets kind of, it enlarges, and then it sort of keeps folding over and doubling back and creasing and folding. And that gives it a unique shape. So here we are, week 5. We're already starting to see these folds develop. You can start to see the cerebellum forming. You start to see the cerebral lobes beginning to form, a little hint of the eye. And now here we are, 13 weeks, end of the first trimester. So if you have kids, or you're familiar a little bit with it, here we are into the first trimester, just very, very tiny. We're talking about a couple of ounces. But you see, the brain is pretty well organized already. And you can see these structures, start to see the parts of the eye, even though they're non-functional, the brain. So it really demonstrates the human brain. And lastly, and I'll just flip back to this, here we are at birth, and you can see the full-blown picture. All the folds that are there, these are called, the indentations are called sulci. A sulcus is an indentation. And gyri is sort of like an outpouching or a mound. And some of them have very, very specific names because they're so important. And they divide things. You're going to see that we divide things regionally. So 80% of your brain are the cerebral lobes, including the cerebral cortices. And then maybe another 10% is back here is the cerebellum, okay, which is sort of sits behind the brainstem and is even covered to some extent by the cerebral lobes. And everything else, the diencephalon, the midbrain, the pons, the medulla, and this is the start of the spinal cord down there, relatively small. Maybe we're talking 3 or 4 or 5 inches, depending on your size. Almost like a stalk, a little bit larger in the diencephalon, with some very important features, not the least of which is here, giving way to the pituitary gland. So you're going to see a lot of these as I go through this sort of base anatomical discussion of the parts of the brain. Now, back up. So here you can see the adult brain, yours and mine, cerebral hemispheres themselves, the diencephalon. We're going to get into the parts of it. And I have a lot of information on that. It's in the lectures, and I'll include that with you, and I'll make sure we put that out on the board on Wednesday before we do the brain dissection, which is really the part that we're doing Wednesday, and then the following week is the final in any event. Pardon me. And here's the brainstem, and then the cerebellum, sort of unique and separate. So here's that picture you saw before. So what's gray matter? Gray matter is cell bodies. Remember, we don't use the term nucleus. It is the nucleus. But because of the way anatomists describe things, they give nuclei a different name, as you will see. So it's all the interconnections called synapses, where the nerves join together, and the cell bodies, and all the material that's not white matter. And it's really not gray, but they called it gray because it's not white. And white matter is all the connections. They're called axons. It's how they're transmitting lines. So just like you would see a power line or a phone line or a wire connecting your computer to a peripheral unit, now that everything is pretty much Wi-Fi, that those black coatings on those wires, if they were white, that's what white matter is. It's just ways that we transmit and receive things. And the basic pattern around that fluid filled cavity, we have a little bit of gray matter, sometimes little islands. And it's interesting, that's where we give the term nuclei to, and then white matter abundantly external to that. And then only in those selected areas do we begin to see little bits of gray matter on the exterior. And again, those are the cerebral and cerebellar cortices or cortexes. So they give you little cutaways that are in here, which you'll see. And to me, it's just sort of fascinating. This is classic spinal cord. You can see where the slice here, slice here, slice here. This low slice. So spinal cord gray matter looks like a little bit of butterfly. Got that little hollow area filled with cerebrospinal fluid. That's called the central canal or the central cavity. And you have gray matter on either side. These are called horns. And we use the terms dorsal and ventral rather than anterior and posterior, though you could use them interchangeably. And you see only white matter, no additional gray matter, no little islands of it outside of it that are there, just this distinctive feature. That's what defines the spinal cord, whether it's up here at the bottom of the brain, at the start of the spinal cord, or down at the bottom, very similar. The only little nuance are these little bumps here that we see in the thoracic level. These are, so you have, these are called lateral horns. So the gray has a dorsal horn and a ventral horn. And these are called lateral horns. They have little nerve endings that go to the muscles around your ribs that play a pivotal role in breathing. So it's kind of important. But other than that, you see the same thing. Sensory input comes from the posteriorly. Generally, the gray matter goes all the way pretty much to the back. Posteriorly, which is somewhat of a defining feature when you're the distinction between the dorsal or back aspect and the anterior or front aspect. And here you have so these are called sensory horns sometimes because all your sensation comes this way, and all the motor units kind of come out that way, with the exception of these little guys on the side. So when we get these additional bits of gray matter around that central core of gray matter, around the hollow tube in the middle, that's why we start to see the brainstem. And you'll see it very much distinctively different when we get the cerebral hemispheres and the cerebellum. So here, this is another classical area. This is the medulla. So you have a little bit of gray matter, actually a fair amount, surrounding that, a much bigger cavity. No longer is that the central canal. It's called the fourth ventricle. Really important because this is where a lot of cerebrospinal fluid is sort of finalized and released, because you have what's interesting about the brain and the central nervous system in general, the brain and the spinal cord, is it's buoyant, it's floating. It's floating in fluid that surrounds it, cerebrospinal fluid, and is contained, and it's got fluid in the center of it. And in the center of it, that gives it that additional buoyancy. That's what this fluid does. And the way that the fluid gets out to surround it is either way down at the bottom or in little openings. There's three of them, one on either side and one on the top, that are located in this thing called the fourth ventricle. So it's really important. And then you see all this other gray matter. All of these, and they have different names or different kinds of nuclei. And what they do is they are control areas, again, for basic functions like breathing and heart rate. We call those automatic, and the fancy name is autonomic functions, things we don't have any control over. I mean, you could hold your breath, but eventually you don't have enough oxygen, you're going to faint, and then your breathing will resume because you can't voluntarily hold your breath that long. Okay? There's just things like that are there. So all of those controls, sort of the presets that you might have, just sort of like whatever the default settings, if you want to think of it in computerized terms, that would be there. And now we go to a much higher level. Now we're taking a slice through what's called the frontal cortex and the frontal lobes, one on each side. So as you're looking at it, this would be right and this would be left. Now you have these lateral ventricles, which have a big portion here and even a smaller portion here. You have a very deep sulcus here. That's along, this is called the lateral sulcus, and it sort of divides probably the frontal lobe from something called the temporal lobe. And you can begin to see that's another one, that's part of the ventricle here, and even a little tail of the ventricle here. So you have islands of gray matter in here. which are called nuclei. And because of where they're located, you're going to see they're called the basal nuclei. And they play an important role in sort of helping to fine tune motions, if you want to think of it that way. The best way to discuss them is if you know someone or you've seen someone who has Parkinson's disease. Parkinson's disease is a movement disorder. They have difficulty starting and stopping. They move very slowly. Sometimes they're rigid. They're jittery. It's like it's like you see it. Typically, you'll see it more in an elderly person who's trying to reach out for something, and their hand or their arm is shaking. Now, if it becomes so severe, even in people who are younger, typically it's a disease that has a variety of sources: trauma, infection, tumors, and insufficient amount of things called neurotransmitters, signaling chemicals that they don't make enough of in this particular area. And all of these are very, very common comparatively. It's a more common disease than you can think. And so those are the basal nuclei, very, very famous. So your brain says move your arm, but to move your arm efficiently, either quickly or slowly, whether you're grabbing for something that's falling quickly or very, very delicately, let's say you're doing a little project with your hands, even just putting in a screw. And a screw, you have to just sort of delicately, if it's very, very small, something mechanical that you might be doing is like that. And now on the exterior, you have additional gray matter. It's thinner than you think, but extraordinary. It's just filled and filled and filled with tons and tons of connections. So when you're thinking, when you're listening to this, these are very, very active. The ones down here are where you're actually hearing my voice and processing, hopefully, what I have to say. So here are the ventricles, and because of all the folding and enlarging, all that stuff, they're filled with cerebrospinal fluid. The particular cells that kind of help to move the fluid around and sort of finalize it. It comes to some extent from the plasma, but it's not the same. And little touches, different chemical balances are part of it. So these ependymal cells, which are part of the neuroglia, which are the specialized connective tissue that supports the central nervous system, and they have quite an interesting amount of anatomy associated with it. You can see the big ventricles. They have a tiny membrane between them called the septum pellucidum, which, if you're dissecting a human, you can see it with the human brains. We do the sheep brain, you don't see it. And they're connected, as all these structures are, by a thin passageway called a foramen, just like a hollow opening. And that's between the ventricles down to the third ventricle, finally to the fourth ventricle by something called the cerebral aqueduct. Aqueduct in the whole ancient Rome, when they were water passageways that they very advanced with channeling water supply, were the Romans who really developed these structures, as you can still see them in, remnants of those from ancient Rome, with the very, very advanced culture for, something that was 2,000 years ago, and there's plenty of them. Just all that, it's just remarkable when you think of what folks were able to do with construction and materials, the pyramids, for instance. Oh, it's remarkable. All the way down to this thin opening spinal cord. So the fourth ventricle, very large. And what you're going to see is that there's additional openings in that fourth ventricle that allow fluid. And we're going to go over what's in the meningeal areas and how the fluid kind of goes from being inside to outside. And this is a good look-see. So here are the interior ventricles. Now understand, there's a whole nother layer of coverings around the brain called the meninges or meninges, and within them, in the center of those, is all of this. It's a passageway where this fluid escapes through these openings here and one kind of back here, called apertures, that escapes and surrounds the exterior. I mean, it's floating. So you can see these big wing-like C shapes there, and there's the divider, the pellucidum, and you can see how they're C-shaped and pointing back. So it's frontal lobe, parietal lobe, occipital lobe, temporal lobe. Here you can see them sort of centrally placed, like it looks relatively flat here, but you can see the shape as it's sort of incorporated and surrounds the area known as the diencephalon. Then you can see it It looks narrow here, but very, very wide. Here is the fourth ventricle, and then all the way down to the central canal. And we see, as you can see, more than 80%, the big cerebral hemispheres, gyri and sulci, fissures. As you can see, the one that follows the sagittal plane that we talked about earlier, dividing you right and left, there's a big fissure. And inside that fissure are blood vessels consistently that collect the cerebrospinal fluid and venous blood and sort of put it together and collect it in a very elaborate, and you look at that in AP2 lab, called sinuses that basically send it back by way of the jugular vein and back to the heart for oxygenation. We recycle everything. And there's a transverse cerebral fissure that you can see on, and you can see this on the dissection. You'll see this on the dissection. There's a lot of them that you'll be able to see that separates the cerebrum from cerebellum. So this is that long fissure here, and the other one you'll see better on the dissection. All of the sulci and gyri have these little veins that are incorporated. And so, unlike bones, we don't necessarily see distinct divisions. And because the sheep brain is probably a quarter or a third of the size, maybe even less, maybe I'd say, dare say, a quarter of the size of our brain, we don't see a lot of anatomy associated with it. So, what we have is the frontal lobe and the parietal lobe. This is all motor and reasoning and thinking and calculating and language skills and communication. This is sensory, purely sensory. If a bug got on your arm, you'd know it and you'd feel it here. Okay, if you stubbed your toe, you'd feel it here. That's where the feeling comes from. Occipital lobe is about vision. Temporal lobe, mostly hearing, but a lot of other things, hearing, a little bit of taste, a little bit of smell, also comes in there. There's sort of a mixed lobe. There's even a lobe deep inside called the insula, which is very primitive, that plays a role in some of those as well. And there's the cerebellum sort of underneath in that fissure you'll be able to see in the dissection. So you can see how they're all divided. So if we go back and look, this is called the central sulcus. And it's really important because where we actually tell our body to move is right here. This is a gyrus, sort of an outpouching or like a hillock, and it's called a pre because it's before the central sulcus, precentral gyrus, and this is the post-central gyrus. This is really where we feel that when you stubbed your toe. This is where that pain fiber went to. That's there, and you're going to see a map of it, very interesting, called the homunculus. But there, let me go back once more. You can't even detect where that is on the sheep. You can only kind of guess. This is called the lateral sulcus, and that's pretty distinctive even on the sheep, where you can tell where this temporal lobe is. But you can't see the divide here at all between the parietal and the occipital. So they're named for the overlying skull bones, as you can see. And here's the ones I was telling you about. So they've done it here with blue reflecting sensory and the reddish color, red or pinkish kind of color, reflecting motor. And so here's that central sulcus, and here's the pre and post central. So because we dictate our movement from here, this is called the primary motor gyrus, and because we actually feel a lot of our distinct sensation from the body here, this is the primary sensory gyrus. So you can see the frontal lobe is the largest and really does the most activities that you can see there. And so they're just trying to show you the distinction. You can see the gray matter underneath it. It's there. If it gets noisy in here, there's a pooch who conceivably could be barking. So, and there's this other layer that really isn't that important anatomically for us, called the insula. It's just a primitive layer on the interior. So you can see, and there's a lot of more specific areas that you can see. But typically, when we look at the cerebral hemispheres, we have the cortex on the outside, then white matter, and then these basal nuclei that are much more deeply placed. So when we think about the cerebral cortex, they talk about it, they call it the executive suite. It's just the highest order functioning stuff. It's you're aware from here. As long as that's functioning, when someone is in a coma, that area is really not processing information. There's been some kind of damage. For whatever reason, signals are not getting through. When it talks about sensory perception, I mean, that's when whether I just touched something on my toe or whether I stubbed my toe. Voluntary motor, making a note, saying something, communicating, developing the words, putting them in the right order, picking the words that allow you to figure out what you're going to say. A ton of memory storage. Memory's all over the place, but high, very much in this area, and certainly comprehension. It's thinner than you think. So you're talking about something that's probably between an eighth and a quarter of an inch, but a ton of processing, but without the axons. These are just closely intertwined cellular elements, and very, very heavy. When you're thinking that 40% of the mass of the brain, pounds of material storing information. And so what they can do with advanced imaging, like the PET scans or MRI scans, you're able to kind of how the function spreads. So for seeing, we know it's in the occipital lobe. Hearing, mostly temporal. Speaking, frontal lobe. It's a very important area called Wernicke's area. Okay, this is Broca's motor area, and this is called Wernicke's area. This is hearing and processing to understand, and this is formulating and directing speech or communication from here in this Broca's area. And when you're thinking, you do everything. It's all over, and that's where the color coding kind of helps you. So we have motor, movement, sensory, okay, and then everything else is association. Most of your more than well over 90, maybe even 95% is association areas, processing the information. It's easy to take your pencil and mark an answer on an exam. It's easy to, turn your directional signal on in a vehicle comparatively. And you can see something and see if it's a red light or a green light, whether I have to stop the car or whether I have to go. But the ability and to do it in the proper order and the amount is all the processing. So it's mostly association area. And by and large, with some limited exceptions, particularly in the cerebral cortex, the right side controls the left side of your body. So for most of us who are right-handed, as about 90% of us are, the left side of your brain is doing the bulk of the work. And depending on when lateralization can occur only in one. So as you can see, involves an entire cortex in one way or another. So there's a certain amount of overlap that does occur. The right side certainly knows what the left side is doing. It's just sort of interesting in how it all sort of shapes itself. So the primary motor areas, like I said, that gyrus right in front of that central sulcus is really where motion occurs, where you're that's the signal going to your finger or your toe or your arm or your leg. Planning it, getting it ready, giving this preliminary signal comes from the next one called the premotor cortex. Broca's area is really because we're going to express ourselves, so that involves processing a lot of information. And there are eye fields directly in front of it, so it has to do with visual as well as other. So here's Broca's. And on the sensory side, here's Wernicke's. These are commonly affected by people suffering a stroke. A stroke means there's been damage to the brain because of problems with blood supply. This is a motor one. This is a sensory one, and it's the great difficulty with these is, and we see motor areas are more sensitive to lack of oxygen or to blood vessel damage. And so most of the time we see patients who have a stroke, and you may know somebody who's had one, have a very difficult time communicating. They can understand what you're saying, but they can't form the words or write down the answer. It takes a lot of rehabilitation. When it's a sensory stroke, they'll hear the question. They just won't understand it. You'll say, is the sky blue today? And they'll say, yeah, today's Tuesday. However they're going to say it. So there's a tremendous amount of areas that are here, as you can see, in the frontal cortex, multitasking, object recall, task management. What do I do first? When do I do it? Spatial tasks. Am I reaching for something up up on top of the counters or on one of the shelves? Am I stepping on the gas? How hard? All those types of things. And the same thing here. You can associate these with messaging from your body and other sensors, and then decide really finally pry here. I'm feeling something now. Did I stub my toe, or I just is my shoe a little tight? That type of stuff. And am I getting signals from taste or from balance? All of those things. Here I am where I actually see an image, but here I am processing what that means because our images kind of come in upside down and backwards. So we have to decide what they are and then put what the colors mean, all of those types of things that are there. And the same with hearing and all processes. So the cells are called pyramidal cells. They're they're the ones that are the primary movers. We call them the pyramidal tracks, and we would look at an area called the pyramids in the body that are really useful. And what they do is precise, skilled skeletal movements. So you'll hear in medicine we talk about extrapyramidal symptoms. That's like somebody gets the shakes from a medicine. Okay. Normally, pyramid is just prime, just making movement. So when you're reaching out to touch something, your pyramidal cells do this. Those are the primary motor cells. They go by something called corticospinal tracts, which means from the cortex. In the cerebral cortex, the motor cortex or frontal lobe, down to the spine, and then from the spine out to the appropriate muscles, and we can map them. And again, it's contralateral. So if this is look at the left side of your brain, it'll be the right side of your body, and vice versa. And so we have one for motor and for sensory. And what it tells you here is much of how we communicate, how we interact with the environment is from our head area, speaking, upper extremity, a little bit lower extremity. So it gives you the proportionality. We're very facially and upper extremity dominated. And when we sense the environment, well, we sense our whole body plays a role in it, but mostly with our hands, our whole face, maybe some with the teeth, very little with our. So when you have a stomach ache, it's very hard to tell. If you ask the next 10 people who have a stomach ache, point where it hurts, they'll point to 10 different areas because you can't isolate it. But if they ask you, you know, what you have a blister on your finger, where is it specifically? They will point right towards it. Where did that bug bite you? Right at it. So as you can see, there's a certain amount of planning that goes on. Patterns are important. Again, this is Broca's in the eye field, and you saw this illustration before. So imagine if we're looking at the inside. It's like, like the brain is you, and we'll do this in the lab. If you cut it in half the long way and open it up, this is kind of what you see. So you see all these motor and sensory areas, all these cortexes, and here's the white matter in the middle. This is called the corpus callosum, which means hard body. This is the piece of white matter that connects right and left, and it's so important that's there. And so you have additional bits of white matter all over that are able to play connectors. And this is an important gyrus, shaped like a letter C, so they call it the cingulate gyrus. And interestingly, so close to this area where the signals come across, there's a lot of it plays an interesting role in emotions. I can't really explain to you why. Uncus plays a role in smell, for instance. So there's a lot when we get into detail anatomy. As you can smell, which is our most primitive sense, okay, that goes directly into the brain. So, and this is when they're talking about opposite side. So you can determine where things are. Sensory, lots of different association areas that are involved. We have almost, there are sensors in almost everything, in your muscles, in your joints, in your tendons. And it's the type of thing where if you close your eyes, you know exactly where your legs are, if you're standing up, if you're sitting down, if you're sitting in a car, if you're lying in bed, you'll know exactly where you are. These are called proprioceptors, tiny mini sensors that tell your body where it is. When those don't work, it usually means there's some type of disease entity that's going on that's damaging the circuitry, if you will, going up. And the same thing we saw before. And then vision is very complicated. I don't spend a lot of time other than to tell you it's in this occipital lobe area. And the same thing with sound is mostly in the temporal lobe. And balance and hearing go in together. It's again, balance is even a little bit more primitive, so that insula, which is very primitive, plays a role in this, and it's really about your head position in space. Smell is also even more primitive, so we see it in the temporal lobes, but very, it's the only sense that's really processed directly in the upper areas of the brain. That's why smelling salts work so effectively at stimulating you. In other words, waking you up or breaking you out, when somebody's dazed or things like that. And taste is also relatively primitive as well. And we saw those before. Obviously, falling on the back of your head, people very often have problems with vision. And now you're beginning to see these multiple areas play a role as well. And I don't want to go into all this. That to me is more. Lastly, is there something about the limbic system? My dog is getting antsy. The limbic system, which involves that cingulate gyrus and a couple of other areas that we'll go over, plays such an important role in emotions. Emotions are part of us. So sometimes we react to things emotionally and sometimes we don't, but we need the emotions to really process things appropriately. So I'm about done. The lateralization is between the two. And again, 90% of us left side dominant. Typically, these are good, and 10% about right side dominant. So people who are left sided, language, math, logic, I'm very left sided. Right sided people in general, and then there's a wide variation with regards to this. You can't paint with a broad brush. Artistic, musical skills, which I have none, very emotional behavior, which I typically don't have. And that's all for now for this little presentation. I will get back to you and I'll send this to you and I'll have some more to do and send you today. Thank you.